Mycobacteriophages? There we go. All right, so the first question we need to answer is, what is a mycobacteriophage? Mycobacteriophages are sort of a subunit of the overall group called bacteriophages. Bacteriophages are, as we've already established, viruses that infect bacteria. Mycobacteriophages are those that will infect mycobacteria in particular. What is distinct about mycobacteriophages is that they have two distinct life cycles. The first is the lytic life cycle. This is where they will insert their genome into their host bacteria, and they'll sort of hijack the DNA replication, transcription, and translation systems. That is how they can build the phages inside of, or the viruses inside of the bacteria, and they'll lyse to bursting the cell and then killing the host. The second type of life cycle is the lysogenic life cycle. This means that they're going to insert their genome into the DNA of their bacterial host. So then the bacteria can continue to replicate and live happily ever after, as will the viral DNA. And if it undergoes stress, the virus realizes, I need to get out of here. That's when it will then go into the lytic cycle. And then what can happen in both of these is we see examples of mobile genetic elements. That just means sections of DNA can go between the phage or the host, and it can be bidirectional as well. Now onto the fun part, the materials and methods. Since we're all part of the SEA-PHAGES program, we're already pretty familiar with the isolation and annotation process, but we're going to focus on the annotation process for now. So our phage was phage Dublin, and we took the FASTA file, put it into DNA master, and let it auto-call the genes and such. So then we went back through those auto calls and then double-checked them. We did this in a few ways. The first way was with GeneMark. So we double-checked to see if DNA master matched with the coding potential that GeneMark mentioned. And the second was StartRater. If you aren't familiar with StartRater, that lists the different start codons that a gene could have, and then lists which would be the best one. Once we had established where each gene started and stopped, we then figured out what the job of each single gene was. And of course, our favorite, Family-Reader, yay. So that tells us the job of each gene and then the family that it falls into. Phages DB BLAST-P had a very similar function. We took the amino acid sequence for each gene, plugged it into BLAST-P to see if there were any homologous proteins. And HH-PREB functioned the same way. At the end of this process, we created a phylogenetic tree using the software Genius. I'll get to that part a little bit later. As you probably recall, as you were going through your annotation process, we had the guiding principles listed by the SEA-PHAGES program. Guiding principle number 3 proved to be the most valuable. The gene density in phage genomes is very high, so genes tend to be tightly packed. Thus, there are typically not large non-coding gaps between genes. Elsewhere in the guiding principles, it also states that we should not expect more than an 80 base pair gap. But as we were going through the annotation of Dublin, we encountered an anomaly. This picture is from the ORF analysis in DNA master on the extreme right-hand side of phage Dublin's genome. As you can see here, we have the second to last gene and then the last gene, with no auto-annotated genes between it. Ladies and gentlemen, this is an 863 base pair gap. And according to guiding principles, this should not often occur. So our research was centered around figuring out why this happened. So the first step we took is we highlighted different regions that we thought could potentially have coding potential. We used the insert gene function in DNA master and then took the amino acid sequence and then plugged that into phages DB to see if there were any homologous proteins. We did not find any. So we thought, okay, well maybe GeneMark will list something inside of the gap. Again, GeneMark did not list any coding potential. So at this point we wondered, okay, does this last gene even exist? Because for the sake of annotation, it would be much easier to say that the genome ends right here as opposed to trying to explain to GenBank why we have such a large gap there. So when we went back to check GeneMark, we noticed that yes, it does indeed have coding potential. So now we're sort of stuck with this 863 base pair gap. So at this point, we took a step back and said, okay, well, how does this compare with the rest of subcluster A5? So this table shows the extremely strong homology that Dublin shares with other A5 phages. For example, phage Thea, which I'm going to call the sister phage of Dublin, shared a 95% identity with Dublin, meaning their genome is 95% the same. This FamWriter map shows us something very interesting. Dublin's not the only one with this anomaly. As you can see here, phages Thea, Cuco, and Little Cherry, all from subcluster A5, all have the same 800 base pair gap. So this is not just some anomaly and some random phage. This is rather a hallmark of subcluster A5. So at this point, we wondered, well, how does subcluster A5 compare to the rest of cluster A? So we took another step back to compare it with other cluster A phages. So at the top, we have phage Nasiatali, which is from subcluster A2. And as you can see, it does not have the 800 base pair gap, but rather behaves in the way that the guiding principles dictated that we would expect. So we thought, okay, Nasiatali looks pretty interesting. We chose Nasiatali originally because when we looked at the DNA master files, we noticed the very similar 4-to reverse gene ratio. And then, of course, obligatory dot plot, as Dr. Hughes mentioned yesterday. Nasiatali and Dublin have a 56% identity, and in terms of what the threshold should be, we would expect 50% in order to make a reasonable comparison between the two, so this has 56% identity. Okay, so based off the fact that we could draw a reasonable comparison between Nasiatali and Dublin, we went back to FamWriter, and as you know, if you highlight a sequence, a certain region, as is here, you can go ahead and get both the nucleotide and amino acid sequence. So at this point, we thought, okay, Nasiatali and Dublin are both in cluster A, which means they probably have some sort of common evolutionary ancestor, and Dublin might have strayed from the pack as it was jumping from phage to phage. Could it be that these regions here might have protein coding regions inside the gap? So we highlighted the regions corresponding with genes 89, 90, 91, and 92 to see if they coded for any proteins. And similar to the last time, there was no protein coding regions there. So at this point, we wondered, okay, are Nasiatali and Dublin actually that closely related that we can draw comparisons between the two? So we created a phylogenetic tree using Genius. Now, in case you're not familiar with the Genius software, you can go ahead and plug your FASTA file into it, align it, and then it'll just go ahead and create this phylogenetic tree for you. So each of these areas show a common evolutionary ancestor that the phages will share. These phages here are all from subcluster A5. Nasiatali, as we mentioned, is from A2. Now, in order for the Genius program to work, you have to have outliers that are completely unrelated. So we chose Firecracker from cluster O and Kimbote from Q. And as we can see, Nasiatali and Dublin do have a common ancestor. So yes, it is still reasonable to draw comparisons between the two. So where does this leave us? Phage DNA likes to jump around with the phages. And what is possible is that as Dublin was evolving, it might have lost some of its DNA to a bacterial host, or there might have been the insertion of start codons that might have interrupted the translation process of some of the genes. We also know that this gap is common in subcluster A5 phages, though it does not have any coding potential, and Nasiatali and other A5 phages do have a common ancestor. But we're not done yet. So in terms of future research, we want to sort of step away from the strict bioinformatic analysis. What we want to do next is to design specific primers for genes 86, 87, and those regions in the 800 base pair gap where we would expect to see protein coding regions. Then we would add reverse transcriptase, nucleotides, and heat-resistant DNA polymerase in order to make cDNA. With the help of reverse transcriptase PCR, we can then replicate that cDNA, perform gel electrophoresis, and then identify if there are potential transcripts to be found in that 800 gap region. Of course, I did not do this research alone. Madeline McAllister, a fellow LeTourneau student, was the one who isolated, purified a characterized phage doublet back in the fall of 2015. Our thanks goes to the Howard Hughes Medical Institute and the Pittsburgh Bacteriophage Institute for hosting the SEA-PHAGES program. Thanks to my lab partner, Junia, who was able to help me with the annotation and research project. And of course, Dr. Balraini and Dr. Frederick for their endless help and encouragement throughout the process. And with that, I am open for questions. Thank you so much.
Questions for our speaker?
Yes.
So when you were looking at the gaps and looking for maybe other things, places where that might be found or whatever, did you ever take the look at the family that that very last gene is in and see if there's other phages that have it and where is it located in those genes?
We did not. That was a really good idea. That's a good idea. You should try that. Yes.
You mentioned some other cluster A phages that were not A5s. I was wondering if you happen to look at any A4 subcluster phages to see if they have this gap.
We have not done that yet.
Okay, so the reason I'm asking is, if you remember yesterday when I was showing a bunch of bar charts of gene expression
Yes, I
data.
remember.
Okay, so one of those was phage Campy, which is an A4, and I only highlighted the expression of the genes that were called, but it was an RNA-seq experiment. So there's a data set for gene expression in the whole genome, and if that gap region is in the A4 genomes as well, you could just look at that data set to see whether it's highly expressed. And I can tell you that in some other phages, like Giles, which is not an A cluster phage, there are regions that are non-coding that are incredibly highly expressed, much more highly expressed than any of the protein coding regions within the genome, and no one knows why.
Interesting.
So it might be a good thing to look at.
Yeah, it would be. Sure. Yes, ma'am.
Did you do a nucleotide blast?
On this region? Yes, we did. And there was no homologous proteins.
I mean, homologous nucleotide?
No. Correct. There were no homologous nucleotides.
Just a real brief. So you may not know the answer to this. You guys, I don't have my phone. I can't look it up. So are the A2s the closest relatives to the A5s on the split streak? I don't think so. Because you might want to look at a different subcluster of
Yes.
A's, like Dr. said. But yeah, in addition to the A4s, I've got it somewhere. Let's thank our speaker again.